Okay, so now let's move to the sources of calcium for a smooth muscle cell. Here is just a rough structure of the smooth muscle cell with its tapered ends, and you can clearly see that in place of the deep invaginations of T-tubules that we saw in the skeletal and cardiac muscle cells, here smooth muscles have smaller invaginations, shallower invaginations, which are actually called caveolae. And they have less developed sarcoplasmic reticulum. And you can imagine that the source of calcium, therefore, in smooth muscles is not mainly from sarcoplasmic reticulum, but in fact, the extracellular calcium ion concentration plays a huge role in smooth muscle cell contraction. Okay, so what happens is, we know that smooth muscles are stimulated in various ways, but if we look, for instance, at how nerves stimulated, let's just say this is an axon terminal in close proximity with the smooth muscle cell. It has these random bulbs in the axon terminal called varicosities, which have these neurotransmitters. So varicosities, and these are just spread diffusely throughout the smooth muscle cell, whereas in skeletal muscle cells, you had one axon terminal forming a neuromuscular junction roughly in the midpoint of the skeletal muscle cell, whereas here you have them all over the place, diffusing, allowing the diffusion of neurotransmitters into the synaptic cleft over here to allow action potential to form, or sometimes smooth muscle cells do not have action potential. They could have graded potential, or what's called junction potential, to flow through gap junctions. But overall what happens is the propagation of any form of potential or depolarization, again, triggers these receptors on the caveoli surface. Again, the DHP receptors, and then you have the ryanodine receptors on the sarcoplasmic reticulum, and there's various ways in which you get calcium ion source inside the cell. They can be just like in cardiac muscle cell, the calcium-induced calcium release. You also have secondary messengers triggering the release of calcium from the sarcoplasmic reticulum. And examples of this is a molecule called IP3. And then there's also something called store-operated calcium channels. And what this basically is very interesting. The cell has a way of signaling the intake of extracellular calcium ions into the cell when the amount of stored calcium ions in the sarcoplasmic reticulum decreases. When there's less calcium ions in the sarcoplasmic reticulum, it somehow signals the intake of extracellular calcium ions into the cell, and then that affects contraction of the myofilaments that are attached to the dense bodies and cause contraction.
Okay.
So if we have a source of calcium ions to allow contraction to be regulated, it's also important to remove the calcium at some point so that the muscles can go back to being relaxed out of their contraction state. So there's different ways calcium can move. They can be pumped back out of the cell. They can be pumped back into the sarcoplasmic reticulum. And they actually use what's called the SERCA, which is a pump that stands for sarcoplasmic and endoplasmic reticulum calcium ATPase. So it's an active transport of the calcium back into the sarcoplasmic reticulum and also back out, out of the cell. And something that further attracts this calcium back into the sarcoplasmic reticulum are calcium binding proteins, for instance, calsequestrin, which have a very high affinity for the calcium ions and keep it there despite the high concentration of calcium ions outside of the cell. This affinity that the calsequestrin has for these calcium ions pulls the calcium ions back into the sarcoplasmic reticulum. So that's how it's relaxed, and that's how the muscle can be induced to be relaxed. But something special in the smooth muscle cells, if we go back to how they're contracted, is if you look at this myosin light chain kinase, and if you think of this light chain over here having a phosphate attached to it because this kinase phosphorylates it, and this phosphate is what activates this head to bind to the actin to make it contract. It's important to get rid of this phosphate. To get rid of it, and that is done by what's called phosphatase, which is a general enzyme that gets rid of a phosphate from a molecule, and in this case, it's called myosin light chain phosphatase. And now once this phosphate is removed, this light chain is no more activated, and this ATPase is no more active, and this whole cycle that we went through for contraction basically stops, and the muscle, smooth muscle in this case, relaxes. So that's how calcium removal and the phosphatase act together to relax the muscle. But let's quickly go over the sequence, the step-by-step sequence of how the muscle is contracted. So we saw there's different ways in which the different muscles are stimulated. For instance, here we have the varicosities, here we have action potential propagating down cardiac muscle cells and skeletal muscle cells, and then they induce the sarcoplasmic reticulum to allow calcium to come into the sarcoplasm, or the extracellular calcium ions to come to sarcoplasm, and that in turn, in the case of skeletal and cardiac muscle cells, changes the conformation of tropomyosin, opens up these active sites, or the calcium binds to calmodulin, acts through kinases to allow myosin head to bind to the active sites, and then in the end, this binding of the myosin head to the actin active sites causes a power stroke and ultimately the sliding filament mechanism, which overall shortens the length of the contractile unit of a muscle, and that is how muscle contraction occurs.